So let's have a look at specimen set 2 for DNA, proteins and amino acids. Let's have a look at this question, 6.2. So this is about chromatography. So when you split a protein up into its amino acids, or when you take some samples of amino acids, then you take a piece of paper or something called thin layer chromatography plate, which is like a glass plate with a fine sort of layer of, I suppose, gel you'd call it on it, but the principle's always the same. You dip the bottom of the plate or the paper into a solvent. You put a start line with pencil, not with a pen, because the pen would also split into sort of different colours in the solvent and would dissolve in the solvent, so you do it with pencil. You put the start line above the level of the solvent, so the solvent doesn't go above the level of the start line, it always goes below. Otherwise the amino acids that you spot onto the start line would just dissolve straight into the solvent. Let the solvent rise up the paper and it splits the amino acids up. You measure the distance travelled by the solvent to the top, towards the top. It can't go above the top because you need a line at the top to be able to measure something called the RF value in a moment. So important that it doesn't go beyond the top. And what actually happens is that the different amino acids separate. They separate based on how well they dissolve in the solvent and how well they stick or adhere to the plate. If they dissolve very well in the solvent, then they rise up the plate or the paper more quickly than others. So if they dissolve well, they go a long way. If they adhere to the plate, stick to the plate, then they don't go very far. If they don't adhere to the plate, then they go high up the plate. The solvent that's rising is called the mobile phase, so if they dissolve well in the mobile phase, they go a long way up the plate, and the stationary phase is the paper or the thin layer chromatography plate, and that is the stationary phase. And if they adhere to the stationary phase, then they don't go a long way. If they don't adhere to the stationary phase, then they do go a long way. So I'm looking at N. I'm looking at N's gone further than M. So it's clearly soluble in the mobile phase, in the solution, in the solvent rather, that it's been put in. It dissolves well, and it doesn't adhere well to the stationary phase. So it dissolves well in the mobile phase, in the solvent, and it doesn't adhere well to the stationary phase, which is the paper or the thin chromatography plate. Things that are polar with delta pluses, by the way, tend to dissolve well, but they do tend to adhere. So yeah, you just got a little bit of theory then. It's based on the polarity of the molecules. Polar molecules with delta plus, delta minuses tend to dissolve well, and also tend to adhere. So let's work out the RF values. How do you do that? Well, you measure to the middle of the spots. You measure to the middle of the spots from the start line. And you measure from the start line to the distance travelled to the top to the solvent front. And an RF value is always less than 1. So you do the distance from the middle of the spot down to the start line, and you divide by the distance from the start line to the final solvent front at the top. And you've got some numbers there. So it tells you those RF values all less than 1. So I'm looking that something like M has got a low RF value, and something like N has got a bigger RF value. Which is which? The dipeptide responsible for spot M. Well, if I measure the distance from the start line to the middle of the spot and divide by the distance between the two lines, the start line and the solvent travelled, that's looking like around about, it says on my answers that it's Seer Allah or Alice. Seer Allah is 0.15, so let's have a look at that. Yeah, it's not gone up very far at all, so 0.15, if you get a ruler out and measure the distance between the start line and the spot, divide by the distance between the two lines, 0.15 seems to be about right. It's asking for N, N is a bigger number. It says when I do my measurements it's allylise, which is 0.55, which seems about right. It's gone about just over halfway up the plate. So the distance between the start line and N divided by the distance between the two lines is 0.55. Okay, fine. And then L, order of the amino acids in the tripeptide. Okay, so a tripeptide has got 3 different amino acids in it. So when it's been hydrolyzed using concentrated hydrochloric acid to break the peptide links, peptide bonds, I've got ser, alla, and allylise. So I've got dipeptides, 2 amino acids, serine and alanine, and I've got another dipeptide, 2 amino acids, alanine and lysine. So clearly the common part is the alanine. So when the tripeptide has been split up in 2 different places along its chain on your peptide link, you've got sera and alanine, and the lysis part has been broken off, and then the other way around, you've got your serine part broken off and your alanine lysis part staying together as a dipeptide. So the order must be serine, alanine, lysine, ser, alla, lys. Let's have a look at DNA then. I've got phosphate, I've got 2 deoxyribose. I always say glucose is bad for you, so draw a cross between it, and then the cross between the 2 shows which 2 groups join. So the OH on the top of the phosphate joins to the OH at the bottom of the 2 deoxyribose. As you can see, condensation reaction makes water, so you remove H2O. And then the bottom of the phosphate joins to the CH2OH at the top of the left-hand side on the 2 deoxyribose. When you're asked to draw it together, remember to remove the H's from the phosphate that are carrying on up and down, because obviously they will form condensations as well, up and down. We'll have a look at that in a minute. And then you've got the bases. So I've gone through that before on the previous video and said about lone pairs of electrons on N's and O's, delta pluses on H's of NH's, because you've got difference in electronegativity between N and H there, so you're going to get a polar bond N minus and H delta plus, and then they all bond using hydrogen bonds. See the previous video for that. So let's have a look then. So I've got, you can see where the bottom of the phosphate joins onto the top of the 2 deoxyribose, top left, and it's going to make water. I've highlighted H, H and O to make H2O, and similarly the bottom of the bottom left on the 2 deoxyribose with the top of another phosphate. So phosphate comes in top and bottom, releases water in a condensation reaction. Take those 2 H's and O's off each one and then join them together. And as I say, remember to take an H off the top of the phosphate and H off the bottom of the phosphate, because they will be released when they join onto another 2 deoxyribose up and down to form a chain. The chain, by the way, a single strand, is called a nucleotide. Okay, so we've done that now. Where do you join the bases on? Well, you look at the bases and you find the bottom NH on the bases in your data book, and that's the part that's going to join on. So it's easy to identify the part of the base molecule that's going to join onto the OH on the top right-hand side and going to make water as well. So you remove the H2Os from that bottom N on the base, as you see it in your data book. It'll release water. Take them off and join it on. Oh, here I've drawn again how the hydrogen bonds form. So lone pairs of electrons on the N, lone pairs of electrons on the O, and they hydrogen bond around. 3 hydrogen bonds between guanine and cytosine, going around clockwise from top round, next left round to the next right round, next left round to the next right round, and similarly from the adenine to the thymine, top and top, and round 1 and round 1. And it's the lone pairs of electrons on the N's and O's that hydrogen bond to a delta plus on an NH, or an NH2 on the other base. So 2 complementary strands of DNA form a double helix. complete the base pair diagram. So you've got to identify what that is. So and draw the interactions. So the interactions, when you draw them, look like that. So you've got, if you identify that, and we identify it from here, I think we've got guanine there. Let's just double check. 2 ends in a 5 ring, 2 ends in a 5 ring. Yeah, we've got guanine there, so it's going to join, as you can see here, with cytosine. So you draw cytosine there, and then you identify, you put the delta plus and delta pluses and the lone pairs of electrons on, and you join them up, just sketched, maybe with a pencil or something like that, and a rough bit of paper, or to the side or in the back of your booklet, and then put a line through it. And then what you've got to do is you've got to make sure that the hydrogen bonds are dotted lines, but you've got to flip that base, mirror it. So when you draw it neatly in your answer, you've got to flip it left and right so the hydrogen bonds join. Okay, and then you've got to describe it. So describe how they join together with lone pairs of electrons on the O's and the N's bonding to delta pluses on the NH of another molecule. You're just going to describe it. Describe that process there on the bottom, how they hydrogen bond together, what bonds with what. State how the interactions between adenine and thymine differ. So you've got 3 hydrogen bonds between guanine and cytosine, and you can draw that and identify that if you can't remember from your data book, and 2 between adenine and thymine. It says, suggest why urea is effective at separating the complementary strands of DNA. In other words, why it breaks up the hydrogen bonds between the 2 base pairs on separate nucleotides. It's because there's NH2 groups in urea. You can see a structure of urea, and an NH2 group is going to have a lone pair of electrons on the N. So it can bond with a base rather than a base bonding with another base with hydrogen bonds. The urea can hydrogen bond with one of the bases and stops then that base hydrogen bonding to another base because it's bonded to the urea instead.